Okay, hey guys, it's Miss Lynham. We're going to go ahead and do another lecture. Today's topic is going to be cancer. So we're going to take a few moments and talk about cancer in this unit. I know in Chapter 12 we already talked about cancer. We considered it as a set of diseases in which cells escape from the normal control mechanisms that limit their cell growth. So now that we have talked about transcription translation, also known as gene expression, and its regulation, we're ready to look at cancer a little bit more closely and tie it into how different things that happen in transcription translation might affect a cell and cause it to become cancerous. Okay, so the genes that normally regulate cell growth and division during the cell cycle include genes for certain growth factors, as well as their receptors, and in addition to that, molecules of the signaling pathways. So any mutation that alters one of the genes in somatic cells in one of these areas can lead to cancer. The agent of such a change, that would cause it to become a cancerous cell could be random mutation. However, it's also likely that many cancer-causing mutations result from things such as environmental influences, maybe chemical carcinogens, X-rays, high-energy radiation, and then also some viruses, which you see here listed on the notes, on the PowerPoint right here. You can see a few viruses that are listed. This is something that a lot of people don't think about, how a virus may lead to cancer, but the first one you see listed here, the human papillomavirus, there has been a link to people that have this virus and the instance of them having cervical cancer. Of course, this is women, which is why a lot of women and even young girls are now getting the HPV vaccine, which can happen as early as like 14 and 15 years old, and a lot of parents are having their daughters get this to try to prevent them from getting cervical cancer later in life. So that's one virus. It doesn't mean that every person that has ever had cervical cancer has had the human papillomavirus, but the other way can be said, like if you have the HPV virus, there's an increased chance for you developing cervical cancer. The Epstein-Barr virus is another one, and you've probably heard of this if you've heard of mono, because it's the virus that causes mono. And so they've linked that as well, which is kind of scary, to some types of lymphoma. If you've ever heard of Hodgkin's lymphoma, Hodgkin's is one type, and then also there's another type called Burkitt's lymphoma. Those are both different types of cancer that have been caused by the Epstein-Barr virus. This one right here is a virus called HTLV, which stands for human T lymphotrophic virus, which causes adult T-cell leukemia. So having this virus, HTLV, has led to an increase in adult T-cell leukemia. So I just wanted to kind of mention that, because it doesn't always have to be things like carcinogens, pollution or chemicals that are found, like we talked about in water bottles or different things like that. It doesn't always have to be UV rays or radiation. It could be a virus that leads to cancer as well. Okay, so now, next topic is this idea of these things called oncogenes. Okay, there's a lot of research going on right now, and has led to the discovery of certain genes that are cancer-causing genes. These are called oncogenes. Okay, now the word onco in Latin, if you look at onco, it means tumor. So it can be nicknamed a tumor gene. Now there are normal versions of these genes, because not everybody has oncogenes. Okay, so some people possess these cancer-causing genes, which leads to them having a higher incidence of cancer. But there's a normal version of these called proto-oncogenes. You can see it down here at the bottom. But these proto-oncogenes, the normal version, what they do is they code for normal, okay, so they code for proteins that stimulate cell growth and division, the normal cell growth and division. So these are like normally saying, okay, let's go ahead and go through the cell cycle. Okay, so they stimulate normal. I'm going to write this on here in case you're following along with me. They stimulate normal cell cycle, normal cell growth and division. So everybody has proto-oncogenes. Okay, so the question then kind of arises, what would make a proto-oncogene turn into an oncogene? What would make a section of DNA in somebody's body that stimulates normal cell growth and division, what would make that change to become an oncogene and now become like an uncontrolled, gene that has like uncontrolled cell growth and division? Okay, so there's the thing that makes it do that. We do know that there's a genetic change. There's some kind of change that happens to DNA, happens to a gene, which can then increase either the amount of protein product or the activity of each protein molecule. So it's some kind of increase in protein, either production altogether or the activity of that protein. So we do know that is happening. Okay, so there's two categories that we're going to talk about now from here on out in the notes. We're going to do a little exploration into this whole oncogene, proto-oncogene, and then we're going to talk about a second type of gene that also is causing cancer. It's called a tumor suppressor gene. So these are the two ways that somebody can get cancer, the two types, and these are oncogenes, proto-oncogenes, and then tumor suppressor genes. Okay, so I wrote a little bit down here. I don't know if you want to write this down, like push pause and then write this down, but I'm just going to go ahead and read it right now. These proto-oncogenes and proto-oncogenes, it happens like when it changes, it comes from a genetic change that leads to an increase in either the amount of proto-oncogenes protein product or the activity. It's what we mentioned right up here. A tumor suppressor gene, just read this, you can kind of see, tumor suppressor gene. It's a gene that normally produce products, okay, look at this, that inhibit, that means stop or slow down cell division to prevent uncontrollable growth. So everybody possesses these tumor suppressor genes. All of us have them. They're genes that are going to produce products to slow down cell division so that we don't end up with cancer. Okay, so if you have a defect to that, and now you have a defect to these tumor suppressor genes, now you're going to not inhibit cell division, you're going to have uncontrollable cell division. So there are two main ways that you can develop cancer. Okay, so let's go ahead and explore a little bit more. Let me see if I can get out of this little screen right here. Go to the next one. Okay, so here's a closer look at oncogenes. Okay, so the genetic changes that convert proto-oncogenes to oncogenes fall into one of three categories. Okay, you can see them listed down here. Amplification, point mutation, and movement of DNA. Okay, so cancer cells, the more research that they've done, are frequently found to contain chromosomes that have broken off and rejoined. That would fall into this category down here of movement of DNA. Oops, that didn't work too well. There we go. Of the movement of DNA. So if, let's say, a piece of DNA breaks off of one chromosome, okay, you guys remember this from the mutation part. So if you've got a chromosome and a piece of this breaks off and is translocated to a different chromosome, and I'm not talking about crossing over in the formation of sex cells. I'm talking about like a piece literally breaks off and translocates with a different one. Well, if that happens, and if it puts that piece of the chromosome, you can see up here, close to an unusually active promoter, then that means that that gene is now going to go through transcription and translation faster, more often, and we're going to have an excess in proteins produced. Okay, so a movement of DNA can definitely change the rate at which the gene is transcribed. This is the case, and you can see down here, in translocation, breaking off a chromosome. It's also very common in these things called transposons, okay, which are nicknamed jumping genes. It's when a stretch of DNA can move from one location to another within the genome by a type of recombination process. So that's one example. They never completely detach, but are brought together by enzymes and the bending of DNA. So this is a common thing that occurs in some individuals moving that DNA, which could lead to a gene becoming a proto-oncogene, from a proto-oncogene to an oncogene. Okay, another way that this can occur is a point mutation. Okay, point mutation in the promoter for a proto-oncogene or in the gene itself. Okay, you can see it over here. So what you have is a hyperactive or degradation resistant. In other words, it's a product that resists being broken down, or it could be a hyperactive type of protein that's produced. Okay, the third example is amplification. This one's kind of interesting because it's like the gene is amplified or multiplied. So you've got multiple copies of the gene. Okay, instead of just having one copy of the gene, there's several, and all of those will go through transcription translation, and so you're going to have an excess in the amount of protein produced by this way as well. So it's just three different ways that you can see a proto-oncogene become an oncogene, and that's through amplification, point mutation, and movement of DNA. Okay, so the proteins encoded by many proto-oncogenes and tumor suppressor genes are components of cell signaling pathways. So let's take a closer look at how such proteins function in normal cells and what goes wrong with their function in cancer cells. So this slide, okay, is called a RAS, is what we're going to talk about, the RAS gene. And in a minute we're going to talk about the P53. Okay, so basically we're going to focus on the products of two key genes. Okay, this is the RAS proto-oncogene, and in a second we're going to look at P53 tumor suppressor gene. Okay, so listen to the terminology. This is RAS proto-oncogene. Okay, oncogene. So we're going to look at this one. Okay, so proto-oncogene is different, and in a second we're going to look at one called P53 on the next couple of slides, which is a tumor suppressor. So these are the two categories of the two different ways that cancer can arise in people. Now, just so you know, this is a side note, not that you have to memorize this, but what I'm about to show you with this RAS proto-oncogene, this basically accounts for 30% of human cancers. Okay, so 30% of human cancers fall into this category called the RAS proto-oncogene. So what happens is a pretty good picture you can kind of see. Here's the cell membrane right here. Okay, so that's the cell or plasma membrane. And then these little purplish looking things, these are receptors that are in the cell membrane. Okay, the little circles that you see coming up here, these are growth factors. So normally, okay, let's just talk about normally. Normally, a growth factor would be the signal molecule, would come up here to this receptor. Okay, it would go and bind to the receptor site. That would be the stimulus, which would then initiate this thing right here called RAS. Okay, now this is RAS is a protein. If you want to know where it comes from or where this RAS gene is named from, just FYI, it's named for rat sarcoma, which is a connective tissue cancer. That's how they discovered this. But basically, this RAS, okay, is a type of G protein. What it does is it relays a signal from a growth factor. Okay, so here's the growth factor. So this RAS protein is going to relay the signal to a cascade of protein molecules. Cascade means like this, flowing one after the next, after the next, after the next. These protein molecules, you've heard these before, called protein kinases. Okay, so the RAS G protein is a relay molecule. So he receives the signal. The stimulus is the growth factor binding to the receptor site here. Then that signals the RAS protein. He activates, passes it to these protein kinases, which then stimulate the signal down here for transcription factor, which is an activator. That then activates gene expression, goes to transcription translation. We eventually get proteins that stimulate the cell cycle. So this product down here, this little triangle, is going to have an effect and cause increase in cell division. Okay, so this is a normal operation right here, is what you see. Okay, how this becomes an oncogene, changes from a proto-oncogene to an oncogene, is right here in this box. There's some sort of mutation that occurs. Okay, the mutation occurs to RAS, which means it no longer needs the signal molecule. It doesn't have to have the growth factor. You can take this out. Okay, I'll just take it out like this. But it's now hyperactive, and it's going to continue to cascade down to these protein kinases. So we're going to have down here our product, increased cell division, is going to occur without the need of this growth factor. That's not good. Because if you have more increased cell division and you're not getting the signal from the growth factor, that could then lead to too much cell division, which could result in a tumor or even cancer, and of course it could be metastasized or whatnot. So that's one main way that cancer is caused, is by this RAS proto-oncogene that turns into an oncogene by having a mutated RAS protein. Okay, so that's one way. Okay, so in addition to the RAS proto-oncogene, another way that cancers can form is by tumor suppressor genes. Okay, so we're going to talk for a minute about these. Tumor suppressor genes, like we mentioned earlier, they encode for proteins that, here's the key word, inhibit cell division. Okay, so these are genes that are supposed to slow down cell division, and therefore any mutation that inhibits activity of one of these can lead to cancer, can lead to tumors. Okay, so it's kind of hard to think about this because you're like, whoa, you know, it's kind of hard to think about tumor suppressor genes, but everybody possesses these type of genes. So when these genes are functioning normally, they're going to slow down cell division if need be. So if you've got some sort of mutation to a tumor suppressor gene, this is what it leads to right here: abnormal cell growth and formation of tumors. Okay, so how do these work? If this tumor suppressor gene is working, it is doing its job, it's going to produce proteins, okay, that not only inhibit cell division, but it also produces products that can repair damaged DNA. So DNA won't be able to, have like mutations occur to it. If it has a damage, this type of gene can actually fix it before it does anything, detrimental. Not only do they do that, but these tumor suppressor genes can also control density-dependent inhibition. You guys remember that, where cells only divide until they're touching other cells, and once they get to that point when they get signals from other cells, they don't divide anymore. So these types of genes control that. They also control anchorage dependence. If you remember, a cell must be attached to a substrate in order for it to divide. Okay, so if we have some sort of, imagine this, a mutation to this gene, we no longer can repair damaged DNA. So the DNA is damaged, and if the cell divides, it now has more cells with damaged DNA, aka cancer cell. We now lose the ability to have density-dependent inhibition, which means cells are going to touch other cells and just keep dividing, and make a big tumor. Cells are no longer being regulated, meaning they don't have to be attached to a substrate necessarily. They can be in the bloodstream somewhere and start dividing uncontrollably. And so these are all things that normal tumor suppressor genes do. So if we have a mutation to that kind of gene, we now have all these problems. Okay? Now, just so you know, a mutation in a tumor suppressor gene is responsible for over 50% of all human cancers. So contrary to what we just talked about with 30% of all human cancers, this seems to be the biggest deal right here, is more cancers are caused by tumor suppressor genes than they are the RAS proto-oncogene. Okay, the gene that is most often defective in human cancers, like they've isolated the tumor suppressor gene, they've actually gotten it narrowed down. It's one that's called P53. You will hear this a lot if you guys go to med school or you're going to go to nursing school. P53 gene is the one that they've identified if you have a mutation in P53, then it causes cancer. What this does, P53, when it's working normally, it produces certain transcription factors. Okay, the transcription factor, what it does is it's going to synthesize, I'm going to write this down, synthesize cell inhibiting, there's the key, inhibiting proteins. So if this gene is functioning normally, it's going to produce factors that can inhibit the production of a protein. That's a good thing. It's sometimes nicknamed the guardian angel of the genome because it's also responsible for controlling a few other genes that we're going to talk about on the next slide. And then it serves as the master brake on the cell cycle if DNA damage has occurred. Okay, so this is the P53 gene. The P53 gene has been called the guardian angel of the genome. Once the gene is activated, for example, by DNA damage, the P53 protein functions as an activator for several other genes. So you can see in the picture, okay, so P53, if it's activated, can have an impact on the P21 gene, which you see listed here. What the P21 gene does is it actually stops or arrests the cell cycle, and it does that just long enough so that the DNA can be repaired. So if P53 is activated, it also has an impact on P21, which can stop the cell cycle, repair the DNA, which hopefully will then prevent, the cell from becoming cancerous. In addition to that, the P53 also activates the expression of a group of microRNAs, which in turn inhibit the cell cycle, and then it can also turn on genes directly involved in DNA repair. So there's a lot of fixing of DNA that can happen from this P53 gene. When DNA damage is irreparable, you can see this line over here, then it will lead to apoptosis, which is programmed cell death. So hopefully one of these routes will happen. P53 can either initiate DNA repair, it can then prompt P21, which can stop the cell cycle and also help repair DNA, or if that can't happen, then it will program the cell to go through cell death. And so it's kind of known as the guardian angel because it can do all of those things, and that's what you see listed over here. Okay, so here's a good diagram of a picture of P53 doing its job. This is a cell cycle inhibiting pathway. So in this pathway, okay, you can see over here, UV light is the damaging stimulus. Okay, so DNA is damaged. See the break right in here? So DNA is damaged. It's in an intracellular signal. Okay, so here's the UV rays coming in, damages DNA. It sends a signal to these protein kinases that you see here, okay, the little green things. So the signals passed via these protein kinases leads to the activation of P53. There it is right there, so it's activated. That activated P53 promotes transcription. Okay, so this DNA is going to be transcribed at a faster rate or at a higher rate. What it's going to do, though, is produce products down here that are cell cycle inhibitors. So it's going to inhibit the cell cycle. Okay, so the resulting suppression of cell division ensures that the damaged DNA is not replicated because it's going to be inhibited based on this product. If DNA damage is irreparable, this P53 signal will actually lead to, ready, programmed cell death. So mutations that cause deficiencies in any pathway component can contribute to the development of cancer. So if we've got some sort of problem with this gene is defective, then we're not making inhibiting factors. If we're not making inhibiting factors, then we're going to have increase in cell cycle. If we have increase in cell cycle, we now have uncontrollable cell cycle, which could be cancer. Okay, so this slide is tumor suppressor genes continued. Geneticists are devoting much effort to identify inherited cancers, okay, which inherited cancers I'm sure you guys have heard a lot about, especially maybe in your own families. You might have some instances of cancers that are passed in your family. The alleles for cancer, if you can test for those, you can figure out the predisposition for certain cancers and detect those early in life. They say that about 15% of colorectal cancers, which is cancer of the rectum or cancer of the colon, are involving inherited mutations. Many of these are tumor suppressor genes, and since only 15% of colorectal cancers are associated with known inherited mutations, research is continuing to identify certain markers that could predict the risk of developing this type of cancer. Another one, in addition to colorectal cancer, in 1990, just shortly before you guys were born, geneticist Mary Claire King, she convincingly demonstrated that mutations in one gene called BRCA1, you can see it listed right here, and BRCA1 stands for BR breast CA cancer. So she demonstrated that in the BRCA1 or breast cancer 1 gene, that mutations in that were associated with an increased susceptibility to breast cancer. Of course, everybody wasn't, they didn't believe her at the time, and of course more research came, and then they identified a BRCA2 gene. So there's 2 breast cancer genes that they have showed that if you have a mutation in these genes, you have a higher susceptibility of developing cancer. They've even put some stats with this, which is pretty scary. I'll read a couple of these to you. But basically, if you have a mutation in the BRCA1 or BRCA2 gene, that is one of the reasons for over half of inherited breast cancers. And then get this stat, if you have one mutant BRCA1 allele, if you inherit that, you have a 60% chance of developing breast cancer before the age of 50. Okay, so all you need is one mutant allele and you have a 60% chance of having breast cancer. Okay, now individuals with 2 normal alleles, meaning you have no allele for this, this tumor suppressor gene, the mutant form of it, you have less than 2% chance. Okay, but the thing is, though, I don't want you to be scared, and because most breast cancers are not the inherited kind. They're not, defects to the BRCA gene. But if you do test positive for it, okay, if you do have the mutant allele, you have a 60% chance of having breast cancer by 50. So what that means is if you do have a family history, you've got a grandmother, you have an aunt, you have a mom, you have people in your family, lots of women who have had breast cancer, then you might want to do the research on getting the DNA testing to see if you test positive for the BRCA gene, either BRCA1 or BRCA2. It's a simple DNA test, a lot like what we did with electrophoresis, where you're looking for the banded patterns, because you can see it show up on when you do this electrophoresis. Okay, we are almost done. The last diagram, and then we're done with this, is just kind of putting together the whole picture, the effects of mutations. So if we've got too much protein or the cell cycle is overstimulated, okay, you can see here in the green, then that leads to increased cell division. If we have increased cell division, that's uncontrollable cell growth, that leads to cancer. Okay, you can also have this occur based on this right side. If the protein is missing that is supposed to inhibit the cell cycle, okay, the red protein's missing, then that's going to lead to increased cell growth. So there's 2 ways that these mutations can lead to increased cell growth. Either we have too much of the product, overstimulation, or we are not inhibiting the cell when there might be damage to it when we're supposed to be stopping the cell cycle. Okay? All right, that about wraps up cancer, and we are done.